ask yourself whether causation really can be inferred from correlation, because we all know that in general, correlation does not imply causation, and that's definitely true for two-point correlation. So my favorite example is between the barometer and the weather. Obviously, there is some association between these two observables, but even though the barometer, tell in advance what will be the weather tomorrow. Obviously, the barometer does not cause the weather, so even time lags cannot really ensure that you have causal relations. So in this example, as we know, the connections really is through some unobserved, I mean, not unobserved, but through some third variable, which could be, for instance, atmospheric depression, to which the barometer responds faster than the weather. That will take some time to arrive on site. So indeed, I mean, neither the barometer nor the weather cause each other. It's really due to some common cause between the two. The surprise is really that when you go beyond two-point correlations, in fact, there are some informations about causal relations between variable when you look at three or more point correlations. So this sort of reasoning and thinking started with physicist and philosopher, I think, Hans Reichenbach in the in the late 40s and 50s, I think, who proposed this very intuitive common cause principle, which states that when you observe two events, E1 and E2, that have no obvious reason to coincide. So when you have this sort of coincidence, in our daily life, we typically infer that there must be some common cause to explain this coincidence. And it usually works nicely. But in fact, in order to apply this kind of reasoning, you really have to have additional prior knowledge about the problem. And you must, in particular, you must be able to exclude that E1 is not actually the direct or even indirect cause of E2 or vice versa. So in fact, there is nothing really that in the data itself that can distinguish between kind of common cause from this indirect effect through some mediator, for instance. So when you don't have background knowledge to exclude that, this is not really operational in practice to analyze data. The real breakthrough in the field came in the realization that the correlation patterns that say something about causality is when you have three variables, two of which are independent, like C2 and C1 in this example, and yet both are clearly associated with the third one, E. So in that case, what this implies is that E, the middle variable here, cannot be the cause of either C1 or C2, because otherwise it would generate associations between these two variables, C1 and C2. So the only possibility is that E receives some input from causes, but usually this three-point correlation is not enough to really conclude that C1 and C2 are causes of E, because the problem is slightly more complex. It's also possible that there is an unobserved latent variable that is, as the weather examples earlier, whether when this latent variable is actually a common cause of C1 and E, or C2 and E. So that's the difficulty in the field, really. But by looking at more and more points, correlations, you can actually sometimes you can actually exclude that there is a common cause between two variables. So you can really infer genuine causal relations that we'll discuss later on. So the network reconstruction is really, the problem of taking some form of data on the right-hand side, on the left-hand side, sorry. For instance, let's assume that they are genes, even though they are not in this example, but this would be, let's say, genes that whose expression you can measure in different experiments, even in different cells sometimes. So it can either be bulk sequencing or bulk expression or single cell expression. So each line would correspond to a single cell. And then that's basically the expression of these genes in these different cells. Typically, when you look at this kind of data, if you have enough data, so if enough rows in your data sets, most pairs of variables, when you take them, at random, they are somewhat associated to some extent. But it really does not mean that these variables are direct regulators of one another, but typically that you have some sort of indirect effect, and the whole idea of this network reconstruction is really to dissect this direct from indirect effect. So you hope that the network that you build will be, able to tell you and inform you on indirect path as well, just by looking at it. So that's the hope. As you can say, so as you can see here, the network is fully directed, and in fact, most methods that try to learn this graphical representation from data have to assume, fairly strong assumptions on the class of graph that they are trying to learn. So, for instance, a fully directed graph like this DAG, so directed acyclic graph in that case. Or sometimes you have other methods that we will discuss briefly that will learn only undirected edges. So obviously there are different methods, and you have to make this assumption from the start. So obviously this kind of method cannot really learn causal relations because they either assume it or exclude it. But there are methods that can actually learn from data this causal relation by using this sort of multiple point correlations that we outlined just before. Another point to mention is that the data itself can be much more heterogeneous. So you can have categorical variables, for instance. And you can also have, in principle, higher dimensional variables like short pieces of sequences, which are not easily classified, let's say, but which may also contain some information with other categorical or continuous variable. So you hope that that there is a need to develop this kind of algorithm to learn graphical models. Another issue, often in particular with biomedical data, is the fact that there might be missing data. So some of these cells have no value. It doesn't mean that this value does not exist. It's just it was not recorded in the table, and that's fairly frequent when you look at biomedical data. So you also want to have methods that can deal with this missing information. So I'll just briefly outline in one slide, so it's going to be quite short, but just these major classes of network reconstruction methods. So the first one belongs to the seach&score methods. So when you are able to estimate a score, so typically related to the likelihood of the model given the data, then you can test many different networks and obviously take as output the ones that maximize the score, and it actually works very well. And the main limitation is that most methods are limited to directed edges. And another limitation is that you can't actually explore all possible graph as soon as you have more than a few 10 or 15 nodes. So you really have to use heuristics to search for good graph candidates, but this has been done. It works very well, in fact. Another kind of opposite sort of method that learn undirected graph in that case comes from this uh, trick that when you invert the variance covariance matrix, uh, you end up finding something that is essentially related to the skeleton of your graph, so some graph without orientation. And that's assuming linearity and assuming some other things. So it's actually a very nice kind of approach too. Its limitation is that it can't work for causal relations. So for instance, if the true graph is the top one, here we see this V-structure pattern that is kind of the signature of causality in this data. So if you have causal data and you try to look at it using this undirected graphical methods, it will essentially add an edge to close this V-structure. Um, uh, and then you have this new class of they are kind of old methods, I must say, from the 90s, essentially, that showed how to actually learn causal relations or exclude causal relations by using yet another type of approach, which are called constraint-based approaches, where you try to find these constraints. So you try to find independencies or conditional independencies between variables that allows you to remove edges. And then by using these multiple correlations in your data, you are able to find that some of these relations must actually be directed, as we outlined earlier. You can also have this third type of edge that are bidirected, that suggest that there was some common cause that is not actually observed in the data set, but that kind of explain the association between some pairs of variables. These methods are, as I said, not recent. Uh, and one of the limitation is that they are really not as robust as, in particular, the score methods to sampling noise. So when you don't have infinite data set, this method tend to suffer from this sampling noise instability. So in my group, because we still think that this method are, on paper great. I mean, in the 90s, there was some great work that proved that, this method actually work in the ideal case. And so our attempt was to try to make them much more robust so that we can actually use them, with finite data set that is all we have typically on with real data sets. So this is what we've developed, and we did it by combining these constraint-based methods with some scoring approach to make them much more robust. So now you can apply the same method to data like single cell expression data that we discussed earlier. And then you can indeed learn that most of these connections are directed. So there are regulators and target genes, and there is a direction. But you can learn it from this data. You don't have to assume it in principle. And then, there are other data sets like clinical data set discuss some examples where you have indeed a mixed type of edges, directed, undirected, and also these bidirected edges that are outlined here with these dots, meaning that there is something, some common cause missing that is not observed in this data set between these variables. So the way we do it, and I'll go into more details afterwards, but just to summarize, the way we do it is by computing probabilities of having our heads or tail at either end of these edges. So now we can end up with edges that have indeed, for which you have a good confidence that you have both a tail and a head. So this would be the, genuine causal relations. But you can also have, edges for which you don't have this confidence of having heads, but rather tails. And then on the other end, you can have, quantitative confidence about having two heads on this edge. So let me show you some example of this clinical network to start with, and then I'll go into more methodological detail so that you can get a better understanding and feeling about the underlying algorithm. So in this first example here, we looked at in collaboration with the neurology department at La Pitié-Salpêtrière in Paris. We look at medical records of patients that were for which typically the family suspecting some onset of a neurological disorder typically in elderly patients. So then the neurologist has to establish precise diagnostics. And typically the question is whether this is more like Alzheimer or Parkinson or maybe some other issue. And so you see that the kind of network that we built is fairly dense for a non-expert like us. It's not really, doesn't look too informative. And for the expert, in fact, most of these edges are expected or well known. But still, the surprise to us is that our collaborator actually liked it nonetheless, because it's not that they would learn too much, but it will give them some visualization of what they already knew from just looking at Excel sheets. They knew that certain variables, and by experience, of course, they know that certain variables are clearly linked, but they didn't have this visual representation, which they actually like a lot, we realized. Then, of course, it would be a bit disappointing for us not to discover any new thing. So, in fact, by looking at all these results with them, they point some, unexpected and unknown associations that were quite strong indeed in our analysis, in particular between this cluster here, which corresponds to the vascular cluster. So these are patients that have had a stroke or even the microbleeding in the brain, in fact, so they may not be aware of it. But still, over time, this leads to memory impairments. And the only way to detect it is by using medical imaging. And so here we have two medical imaging modality. So they're called scales, Fazekas and Shelton's, which were not known to be so strongly associated by our collaborator, at least. And so this corresponds to, so Fazekas, as we saw, is directly linked to the vascular cluster, and indeed it corresponds to this white matter that you can see in the brain, which is basically where the micro or more severe bleeding occurred through the demyelination of the axons there. So that's one aspect, and the other scale is Shelton's that basically measure the atrophy of the hippocampus, which is well known to be associated with memory, and somehow the connection between the two was not known, and so to our collaborator disappointment, in fact, they realized that about at the same time we were doing this paper came out, which basically state exactly the same thing, that there was a strong link between Shelton scales and Fazekas scales. So to us, it was kind of a independent confirmation that this is real. Obviously, it tells, I mean, it's interesting because it makes links between microbleeding and memory impairment, doesn't really explain how how these links works. So obviously, there are all these functional questions that remains, but at least there are some interesting links and avenues to explore from this finding. The second example I wanted to show you concerns a much larger data set. So at Institut Curie, the institute is really specialized in cancer and in breast cancer in particular. So we have studied the different data sets on breast cancer from Institut Curie. I won't show you this data today for the lack of time, but I'll show you some other data sets on a much larger data set on breast cancer that we actually worked quite a bit on. This is from the SEER database, which is the database from the US, where about a bit more actually than a third of people living in the US are registered when they have a cancer. So there are more than 11 million people, I think, in this database. But this is pan-cancer. So here we focus on breast cancer on a reasonable period. So we have still almost 500,000 patients. So that's a lot of patients. And we basically use this data set to try, to probe and to test whether our method, given some real data set, not just benchmark. I'll show you some result on benchmark where everything is synthetic, so you obviously know the underlying model, but this is not real data. Let's say this is synthetic data. Here, this is really real data, and it has probably more interesting characteristics. And because it's clinical data, you have in fact, a very good, background or prior knowledge about most of these connections that you may predict, and in particular, you may know that some features are clearly known to be causes from others, and not vice versa. So you may validate or exclude almost immediately many of these connections. So that's the way we actually used it, and we realized that our methods initially was not performing perfectly, so we had to improve it along the way. But at the end of the day, we realized that when everything is done properly, in fact, this method using multi-point correlations was actually quite reliable in particular predicting causal relations, providing you have enough data, and that's also why we wanted to use a huge data set. So 90% of the causal relations are clearly correct. I mean, they are known, basically. And the nice thing is that the 10 remaining percent are somewhat maybe counterintuitive sometimes, but we believe most of them are also correct. So I'll show you some example. But before that, just let me outline that because of these probabilities to orient edges, sometimes we have we may have a strong confidence that we have a head on one side of this edge, but in fact, we have no information about the other side of this edge. So it could either be a tail, so inferring that it would be a genuine edge, or maybe it's actually another head. So in that case, it corresponds to latent common cause. So, but the data basically cannot tell you in that case. So these are called putative causal relation. So if we look at all of them, but the main problem is when you find a genuine causal relations that really is counterintuitive to what you actually know. And we have a few examples like this, but we sort of convince ourselves that most of them are nonetheless correct. So I'll show you and go with you through uh, this one in particular. So we found, and this is fairly robust. So no matter how you resample your uh, data sets and so on, it's always there. We found that surgery was essentially causing the histological type of the tumor, which is which type of cells are affected by cancer in the breast. Obviously, it doesn't make sense. It's clearly wrong. It's not the surgery that causes the type of cancer in the breast. But the fact is, it's the way this data is collected. So, initially before surgery, the doctor may have, by experience, some good feeling or have some indirect indicator of the type of cancer that this patient has. But clearly, I mean, the precise annotation of the histological type, and sometimes it can be quite complex with a mixture of different types. Um, it can only be done after surgery. So of course, because the data is collected this way, surgery somehow is a major input to the histological type recorded in this data set. And so that's what we find. So we think actually that's it's a valid prediction from a methodological point of view, even though it doesn't make sense from purely causal relations, but that's what is basically stored in this data set. Uh, but I can reassure you, I mean, most of those connections are not counterintuitive like this one. So here, for instance, if you focus on these four variables in green that are related to survival of patients, so you have one survival status, and then you have the cause of death, either by cancer or other cause, and the number of months of survival. Well, all the expected connections and causal relations are recovered. So metastasis is well known to be the cause of death for breast cancer. So that's no surprise, but that's definitely what we also predict. So it sort of confirmed that the approach indeed is able to learn causal relation. There are also some surprises because you saw that the graph is very dense, this time even denser than the previous graph. And this is because we have a lot of data, so we can really pick up very fine and minute effect between some of these variables. But there are even more edges that have been removed. So some edges we don't have them in the network, although we sort of expect them. So for instance, we expect that there is some, because we can show quantitatively that there is some link, there is some associations between the type of insurance of this patient and their survival. In fact, there is almost a twofold improvement in survival when you have good insurance in the US. So this is definitely, this is a very strong effect. This is there. And yet, there is no direct connections. Obviously, it's not completely surprising because it's not insurance by itself that saves you, but the sort of treatment that you can do when you have a good insurance. And this is basically what we found, and we could even quantify. So basically, 50% of the effect between insurance and survival is due to surgery. So this is really the major impact is surgery. Then 20% or more or less 20% is due to chemotherapy. And then there are a few remaining percent that are due to radiotherapy. Then the rest, the 30 remaining percent that are not, linked to treatment, are linked to socioeconomic variables. So depending on, your background and so on, it indeed has an effect on your survival. This is also well documented, but this is what we could recover. So let me maybe now pause for a while and go into more technical detail. So how do we learn this graph? And so I told you that we learn them by combining constraint-based methods or what we call now causal discovery methods and score method or information theory method. So this is the one slide that I'll use to try to explain you this. So causal discovery methods have all in common from the 90s. They're all in common to start from a complete graph. So initially you assume that you need all these edges to explain your data, and then you will basically remove all the edges, as many edges as possible, and typically most edges. You will remove them because you think you don't need these edges to explain the data. And the way you remove them is by finding independence or conditional independence between those variables. So for instance, we'll remove this edge between x and ui by conditioning on w. So this is this relation here. And then so if conditioning on one variable is not enough, then you look for other variables, and you can prune your graph this way until hopefully only a few edges remains, starting from a complete graph. Then when you can no longer remove any edges, meaning that they should be here to explain your data, you can try to find these multi-point correlations in order to orient some of these edges. So you'll try to find this V structure between three or variables. And you orient, for instance, these two arrowheads here pointing towards Z, and these two arrowheads pointing towards UI. So as you realize, this way you may learn, you may infer that some edges have these two orientations. And we think that, so everything is as if there was some common cause in between, as we discussed earlier. So this is the kind of the same correlation pattern. And eventually you may also, once you've learned all these V structures, you may also exclude some orientations. Because if I know these orientations and I can exclude that this is a V structure, then I learn that this is a tail, basically. So using, this data, I can learn both head and tails. But as I told you, in practice, this method on paper is beautiful, but practice, it's really not robust to sampling noise. And the main reason is that, you start very far from the final network. Unlike the score methods where you directly try and score possible networks, and then you keep the ones that have the best scores. Here, there is a very long way from the starting point, which is the complete graph, to the final network, where there are only very few edges from the start that remains. And along the way, you actually make errors. And there is some kind of snowball effect. So the early error, will try to be compensated by more errors afterwards. So at the end of the day, you end up keeping a few edges, but that are not necessarily the correct ones. So that's the first problem. The second problem is that this is very easy to make mistakes because we're trying to learn conditional independence. So we're trying to look at the combinatorics of other variables to explain some edges. And this amount to essentially slicing your data into smaller and smaller bits, because each time you condition on an additional variable, you reslice your data in smaller and smaller subsets, essentially. And so even if you start with a lot of data, by conditioning on multiple variables, you end up with a typically very small subset on which you're trying to find independence between variables. So at the end of the day, it's essentially trying to find the best, or trying to find the slicing of this data that resemble noise, where you have no associations between variables. And because of the huge combinatorics of picking conditional variable, there's always essentially a way to slice your data enough so that it looks random. So this sort of approach have this weakness that you're basically searching for noise, or you run the risk of searching for noise instead of searching for information. So we'll try to avoid that by not directly searching for conditional independence, which has this inherent limitation, but we tried instead to look at information. So we try to find the strongest contributors to each edge that, we don't know initially if we will remove it or not, but, for each pair, X, Y, we start to find the best possible contributor. So that's the one that explains most of this association. And it comes in the form of this 3-point information. So that's kind of a generalization of 2-point or mutual information. And so you essentially subtract these contributions to the initial mutual information for the pair X, Y. And then, if it's enough, it means that you can remove and you have only one contributor, like actually here. So we have only W, but typically it's not enough. So you need to find possibly other contributors that would explain, the remaining part of the association between X and Y. And so you will find a second contributors, and its contribution, given that you already subtracted the contribution of A1, comes into this conditional 3-point information. And then you can go on like this. You don't really know where you're going, but the hope is that eventually you may find some conditional mutual information that is really small, smaller than some finite small number that you can estimate from information theory. In which case you believe that you don't actually need this edge between X and Y, and you can remove it. But you do it with some sort of confidence that you actually subtracted real contributions. So obviously at the end of the day, you'll make mistakes, but you're trying to make the mistakes as late as possible. So initially you only collect those contributors from which you are the most confident. So this is, so you use cause for that. And that's how you delay the moment you have to make a difficult decision up to the end. But at the end, you hope that the skeleton of your graph will actually be more correct than just doing it randomly. And then the nice thing is that this 3-point information, so when they are positive, they are contributions. So you can subtract this contribution from the initial mutual information. But while mutual information is always positive, when you go to higher order, so 3 or more points correlation information, can be positive or negative. And so when they are negative, it essentially means that they are not contributors, and they indeed corresponds to these V structures. But you have a score. So the more negative they are, the less sure you are that you found the more sure, you are that you found a correct V structures, and the more sure you are of this orientation. So that's how we can make decisions in decreasing order of confidence so that we delay the mistakes to the end. Okay, so probably I need to move on. I'll briefly show you some benchmark that shows you that indeed when you combine these constraint-based methods with scores, you improve them compared to the state-of-the-art method that use only causal discovery without scores, basically. So here are three benchmarks. So they are, synthetic data sets from a known network, um, inspired from real life problems, but basically you know everything here, and you can generate, as much data as you wish. And so depending on the amount of data you look at, obviously your prediction will improve. And in these three benchmarks here, we go from a fairly simple example where we have 500 parameters to a more difficult one with 1,000 parameters and very much more difficult ones with more than 100,000 parameters. So you see that our performance in terms of F-score, so F-score means that when you go to 1, your prediction is perfect. So we are not exactly going to 1, but we're quite having good result for this in this simple case. But then, of course, as you can see, the performance decrease when so obviously to get many more parameters, you need many more data, which is the strain that we observe. So as you can see, the classical methods, when the data is the benchmark is fairly easy, essentially catches up. So but they actually need more data. So they are not as good as the method using score to pick the right relation. They need more data, but eventually they manage. The issue though is that they are typically they scale not as well as our method. So eventually you have to stop earlier, and you may not be able to look at so many data. This was some data set for categorical data, and here I show you some results for continuous data, and comparing with other methods that actually analyze continuous data. And what you see is that MIIC actually works, equally well on linear or nonlinear data. Here in blue and red. Red is a recent improvement of these methods, but you see that the trend is the same. While you see that other methods typically, in fact, perform better or similarly for linear data sets, but much less well for nonlinear or truly nonlinear data set where you really have coupling between variables. And this is no big surprise because most of this method actually assume to some level that the data is linear or near linear or simple transformation of a linear data set. So when the things are really nonlinear, they are not as good as expected, but this is really striking. Okay, so let me to finish, let me try to show you some applications on the single cell expression data. So this is an example already a few years back in collaboration with some biology team from Sorbonne University. And in this example, our collaborators wanted to overlay on the same graph both our network reconstruction in red here and the reconstruction from a method that is essentially based on correlation. So as we said initially, you see that in this data, most of the variables are correlated to most other variables. Um, but obviously, these correlations do not correspond to direct connections between these variables. So this is the difference between the rather sparse red network, trying to really distinguish direct from indirect effect from the purely correlation-based network reconstruction. This approach can be scaled to very large network. But for this, you need, in fact, to use prior knowledge, because now with the single cell expression data, you have information about several thousands of genes that you can measure in different cells. But now the methods, in order to scale to this very large network, you actually have to use prior knowledge about the type of genes. And so typically, you will distinguish transcription factors or regulators and target genes that are not regulators. So here we don't have thousands of genes yet, but I kept this example because it's a nice illustration of the fact that sometimes, so this we had only 10 transcription factors and 200 targets here, and you see that in this example, this particular transcription factor cycle in green here is surrounded by latent variable. So this is the cartoon here. And what we found just by chance, but I kept this example because it's quite illustrative, when you add a few more of transcription factors and target genes as well, we notice that all these blue dotted edges that corresponds to this cartoon essentially disappear because we in fact found the latent variable, the missing variable in the previous data set, which is this ABP1 gene. So in principle, as I said, I mean, you can scale this to thousands of genes, but then you can imagine the sort of network you end up with. And we realized by, discussing and interacting more and more with biologists that really what they are interested in, from our experience, is really not to have a reconstruction of huge networks, which are really difficult to look at and interpret. But typically they design their experiments very well with some control variable or perturbations that is well controlled, and two different conditions or more. And they are definitely they're usually interested in one phenotype that they can, and they're trying to connect the perturbation to the output signal. So basically that's what they are interested in. And so they don't really want you to use to reconstruct a network with thousands of genes, but rather they want you to extract from these thousand genes the few ones that few tens or hundreds ones, but not more, that are really relevant for the questions they are actually probing in this experiment. So more and more, this is what we focus on, trying to connect some, experimental condition nodes, basically, that we add in our network to some, readouts, which may be a gene or several genes or pathway. I mean, you can design the readout you'd like. And then, trying to make sense and try to find the genes that indeed contribute to this transmission between the conditions to the output that you're interested in. In this example, that I won't have to go into too much detail, the biological problem is fairly complex. This is to look at the effect of macrophages around tumors that basically control the way immune cells like T cells can infiltrate the tumor and try to attack the tumor. But this is done by remodeling the extracellular matrix around the tumor that the fibroblasts and the tumor cells actually secretes. And in this team, they realized that the macrophages actually have a crucial role in the remodeling of this extracellular matrix by other cells. So you see the biological problem is fairly complex, and obviously it has some very important applications because by changing the infiltration of T cells, you can really improve the immunotherapy that you're trying to design to cure this cancer. And so here, the condition is to, in fact, deplete the macrophage. So when you deplete the macrophage, it has some effect on the remodeling of the extracellular matrix, and in particular, the collagen genes. And as you see, the network, concern very few genes, not the 1,000 ones, although they are all there from the start. And we could outline that there is indeed a fairly simple pathways between the perturbation, the conditions, and the output. And this pathways has been in part validated experimentally both in vitro and in vivo now. Another example that I'll try to describe quickly concern cellular therapy. So in this example, and this is done in collaboration with another lab in Paris at the Necker Institute from the Filatro lab. So they have a very nice model, mouse model of multiple sclerosis. So where you can trigger the disease. So within a few days, in fact, these mice become essentially paralyzed. And what they discovered in this group, obviously was not done overnight, it took years. They discovered that if you inject specific cells that are called Bregs, so they are cells that stably express this IL-10 gene, the mice could be cured within a few days and have a happy life afterwards. So in this project, we were very lucky to be able to analyze this single cell expression data, and Louise, who did that in her PhD, first, in this UMAP, identify the cells that responds to this injection of Bregs, and discovered that one of the major respondents is the macroglia. And so here in this graph, we have this perturbation or treatment node that tells you how this microglia that whose normal job is to protect the neurons, but in this disease, it essentially attacks the neurons. So it's an autoimmune disease. And by this treatment, essentially the microglia are reset to normal protecting state. So it occurs, through a cascading of regulations, starting from this treatment through a number of players, and some of them have been then validated experimentally. We went beyond this now to try to look at interaction between different cell types. Yes, Petra, I see your hand.
Yes, I hate to interrupt you at such an interesting point. Just keeping an eye on the clock. If we can finish in the next couple of minutes, we'll have time for questions. I don't know where you are.
Yeah, I will definitely skip part of it, but I'll stop soon. Yeah.
Okay, didn't mean to. Thank you.
So this is just a brief outlook of the more recent things that we've done when we now we want to go beyond analyzing networks within single cell type, but trying to really try to understand how these cells communicate. And we did that by integrating single cell expression data and cell-cell communication data. So that's one example. We actually did many examples. I don't think I'll have time to detail all of them, but you can actually have multiple, more than 2 cell types. So here it's an example with 3 cell types. You can go to the tutorial of the website also to find out more. This also works for spatial transcriptomic data. Again, I won't go into too much detail here. In this example, that's the infarction, myocardial infarction. So that's a huge data set. So we analyze just a small part of it. And this was done by Oriane in the team, who basically distinguished two types of cells: the ischemic myocardial that will include, you know, vascular muscle cells, endothelial cells in general, and the conjunctive tissue with the fibroblasts and the myeloid cells. And so here the question was to try to understand how these two big class of cells would react to this infarction. So we can build this sort of summary graph, which has a lot of information. And although I won't have time to detail all this, but this is done in detail in the paper. You're welcome to show more details there. But basically, the analysis show that you can recapitulate from the early events, so the hypoxic and calcium shock in the myocardial tissue, to the signaling towards the fibroblast and also macrophages that essentially differentiates to come to the rescue on longer time to try to cure the wound, in fact, related to this infection. And in the very last few minutes, I'll very briefly show you some extension where we can actually do similar things, but by combining, by starting by looking at images. So here, the idea is to couple some image segmentation and tracking algorithm so here we'll try to reconstitute tumor immune systems ex vivo. So in this small, what do you call it, chip, tumor on chip device. And then by tracking cancer cells, immune cells, we try to analyze all this. So the data looks like this. I mean, you can analyze many trajectories, 50,000 images in total. And then you reconstruct some graph that looks like this, where you may have different edges between variables, unlike the previous network that I showed you. And this is because now we start from images and from videos of these cells. So we have information about time. So we can include this information about time and show that there are different effects with different lags. So that's the lags in the unit of a couple of minutes here. Tells you that, for instance, between these two variables, eccentricity and division, there is some association at different timescales. On short timescales, so without lag, or within 2 minutes, let's say there is a positive association. So when the cell divides, obviously they have to become very eccentric right before division. But in fact, a couple of hours earlier, these cells become very round. This is, in fact, very well known by the biologists that when cells make the decision to divide, and this is some sort of hidden variable here that they make the decision to divide, then they need to prepare and synthesize all their materials before division. And during this period, they become very wrong. So this, this approach can really recover this known result, but we could also show new result that probably I won't have time to detail too much, but this is also in this recent paper in eLife here. And with that, I think I will go right to the end, and thanks all the students, PhDs and postdocs that worked on these different projects. I show you some results from Louise in particular, Oriane, Frank, but all the students participated to the study that I showed you. Thank you.
Amazing. Thank you so much. There was plenty there for 2 talks. We have to have you back again, Herve, to spend a bit more time on the applications, which looked really interesting. We have so a few people have written in the chat that they really enjoyed your talk but had to go to other meetings. One of them had two questions, which he said that he's asked someone else to listen to your answers, and then
Okay.
and then we'll catch up. I'll just stop the recording so people feel free to jump in.